First, I want to thank for organizing this, and it's great to have the opportunity to give a summary of what we learned about the use of external data in oncology trial over the last couple of years. Starting point is a meta-analysis in which we looked at design, sample sizes, all the clinical trials in glioblastoma. Glioblastoma is a form of brain cancer with a median survival of about 2 years and very modest improvement in terms of novelty of treatments in the last 25 years. And here, what we have seen is a long list, one per year more or less, of large phase 3 clinical trials, and all of them failed over a period of 15 years. And then we started having hypotheses looking at early stage clinical trials. This is a setting where are very frequent early trials that are single arms, and it's one of those settings in which I think the standard of triggering large phase 3 trials based on single arm clinical trial could be improved simply by augmenting the information that we have and doing analysis that account for external controls that can come from electronic health records or from completed clinical trials. And this is the simplest application, I think, in cancer research, clinical cancer research that we can consider, in which we have a single-arm clinical trial represented in blue. And of course, we want to know what is the treatment. Is there a benefit from the experimental treatment? We can augment the data with, as I said, external electronic health records or a control arm of a clinical trial that's completed over the last 5 years, and then utilize all the methodologies that have been developed in the last 3 decades in causal inference, like matching, to obtain inference on treatment effects. And this slide is just to state one thing. Beyond the analysis of single-arm trials, there's many other things that we can do using external controls, and here there's 2 examples. These are 2 designs that we studied. If you look at the top of this slide, those yellow bars are nothing else than interim analysis, and here we have a randomized clinical trial. Those interim analyses tell us if the data are promising or if the likelihood of a final positive result is becoming so small that it makes sense to discontinue the trial. And what we proposed was a Bayesian model that predict what is the probability of a final positive result based on both the internal data generated from the trial, control arm, and experimental arm, combined with external information. And when that probability becomes too small, then we discontinue the trial. And the bottom is another schematic of a different design, and the main difference is that we also allow for variation of the randomization probability. We don't have anymore just one-to-one randomization probability, but in the late stage of the randomized trial, we can, for example, increase the probability of assigning patients to the experimental treatment, especially if there is evidence of promising large treatment effect. So one of the main points that I want to make in this presentation, we can have a long list of tasks that we try to do better with external data and a long list of statistical methods, but do they work? Does not depend only on the set of statistical assumptions that we work with, the methodology that we use, but it really matters to understand if we have the right external data to do this. So for validation, I don't mean just simulating clinical trials with your favorite probability model, but what we do is experiments that are based on a collection of data sets in a specific disease. And here in the right part of the slide, there's the simplest exercise that we tried. We take randomized clinical trials, and then we look at the treatment effect estimate, interval estimate. We use it as a gold standard, and then we forget for a moment about the control arm, which here is in blue, and we replace it with electronic health records, which is in green. And of course, we try to do it repeatedly for multiple clinical trials. If we see an alignment between inference under the gold standard, so randomized clinical trial, and when we replace the control with external data, that means we are in the right direction. And otherwise, we have to identify what are the distortion mechanisms. And so this is the data collection that we have been working with in the last few years. And we allow other research groups in glioblastoma to use it. And we offer support to analyze their single-arm clinical trial. It includes several studies. What is important, these are all patients, first-line treatment in glioblastoma, all of them treated with the current standard of care, which is temozolomide and radiation therapy. And as you can see from this table, for most of these patients, we have information on all the key prognostic factors, which range from age to Karnofsky performance status at diagnosis and other relevant biomarker in this setting. Here's another approach that we use in order, again, to do validation. And I'm back to the idea of using external data for interim decision during the course of the study. The key idea here is resampling in order to produce with a computer clinical trials. And it's very similar to a popular technique, which is bootstrapping in statistics. We take one group, one control arm of a randomized clinical trial and subsample that allow us to produce clinical trial. If we want, we can spike in treatment effects. And then we use the other data sets that we have as external, separate controls in order to attempt to improve our decision. And then we check whether we have the desired effect of having more frequent discontinuation when there's no treatment effect, and at the same time, we're able to maintain the power when we spike in positive effects. Here's a recent project, again, on this idea of validation. This is a multi-arm platform that I designed a few years ago together with colleagues at Dana-Farber. This is a platform trial that already evaluated 3 experimental drugs in glioblastoma with results published back in 2024. What we did, we said, okay, now that we have this data mature and we know about the treatment effect in this setting, let's try to replace our internal controls from inside, and we use 3 different external control groups. And the main result is that we would have obtained the same inference, nearly identical inference on the treatment effect of these 3 drugs, which unfortunately all of them failed to demonstrate better efficacy compared to the current standard of care. And I complete the presentation just mentioning a few other tasks that we considered. This is a paper we published in Biometrica. First author, Boyuren, is a faculty here at Harvard. And the key task is the following. In oncology, we know that very frequently we have treatment effects that vary across subgroups. And at the same time, we know all of the risk implied by augmenting with external data when it comes to testing in order to make a decision whether a drug should go to the market or not. And here we tried to propose a test that is robust. And here robust, it means that there can be unmeasured confounders or there can be measurement error in the external data, but still we are able to control the type 1 error. And when we have good quality external data at the same time, we have an improvement in terms of power compared to off-the-shelf standard approaches for testing drugs. So here's just the same story represented graphically. Different colors means different groups of patients, and they can have very different treatment effect. And the key question that we tried to answer is there hope for a better power in these studies when we have good external data with a study design that remained robust, because we know there can be all sorts of potential distortions in the external data. And last slide, another paper on a strictly related topic. Daniel Schwartz, another faculty, was a postdoc in my group here at Harvard. And well, this is a problem that in oncology we encounter all the time. We have a lot of randomized phase 3 trials that fail, but at the same time, it takes 5 years or longer to get the data. And when the data are mature, we know more about the biology, we know more about the drugs, and there are many subgroups for which oncologists ask us, well, do we have any encouraging data for this specific group of patients? It happened, for example, for novel immune treatments, as well as several other classes of drugs. And well, here is in this figure just a brief summary of the story. What can happen is that you have zero effect in the overall population per protocol, and then you augment with external data, and you see positive or negative effect on every subgroups. Why that happens? Well, obviously because there's some underlying distortion mechanism. There can be a shift in the measurement of the outcome. There can be an effect of selection mechanism. And what we suggested is adaptation of meta-analysis approaches that realign the subgroup estimates, which are augmented with external data, in order to make them congruent with the estimate that here are represented in red, which is based only on the clinical trial data. So it's a standard analysis. In that way, we avoid publication of a result that is negative for the overall population and completely discordant for every subgroups that we consider, which constitute a partition of the overall population. And this was my last slide. I finished from, I mean, here, of course, it was all about validation of approaches in this idea of combining external data and clinical trial. I have to say, the main difference from when I started studying this in this area and as of today is that initially I was convinced the main nightmare should be a measure confounders. And today, unfortunately, no, there are many other distortion mechanisms that can be severe. One is the censoring of survival. We always consider that assumption that censoring is non-informative as a minor one. Actually, it can be a major one, a major driver of bias when we try to combine this clinical trial data and external information. So obviously, we need to work with a comprehensive list of what can be the distortion mechanism. And here I just tried to indicate a couple of algorithms that can help in doing this task. Thank you.